Hello, my dear students, and welcome back to Victory Batch. And I am your Diksha ma'am. So, today we are going to start with the chapter Breathing and Exchange of Gases, a very important chapter from NEET point of view. You know, there is a trend in human physiology. From every chapter of human physiology, they ask at least minimum 1 to 2 questions. So, why to leave them? Let's get started. So, first of all, what is breathing and what is respiration? You always hear these two words, breathing and respiration. What's the basic difference between them? In breathing, you're just taking in the air and leaving out the air, right? You're just inhaling and exhaling. Like right now, we all are doing like just like this. But what about respiration? With breathing, you have taken in the air and that air have oxygen. And that oxygen goes to your tissues and tissues burn the food with the help of this oxygen known as oxidation and gives you energy. This is what your respiration is. So in the breathing, we take in, we exhale or inhale. Right? It's basically just a mechanical process. You're taking in the air and you're exhaling in the air. Inhale or exhale the air. Whereas in the respiration, it involves oxidation of food. Oxidation of food to produce energy. Fine? Alright, so this chapter is all about breathing and respiration. Now, what are the various types of respiration? There are two types of respiration. One is direct, another is indirect. So, what happens in the direct respiration? This occur in the simple organisms. In which organism? Which are quite simple. Their body is not so complex, guys. Just like we have amoeba. Amoeba is just single cell animal. So in the single cell animal, there are no lungs and no gills, nothing like that. So oxygen will directly get diffused through the body. So in this, direct diffusion takes place from the body. Direct diffusion of gases. Right? Like we have amoeba, like we have the poriferans or sponges. So what happen in these organisms? If this is organism, directly the oxygen will come and CO2 will move out and the cell or the organism will do what it's doing. It's respiration, breaking down the food. Whereas in the indirect one, what happened in the indirect one? The body is complex. Body is complex. Imagine in us. First of all, there will be exchange of gases in the lungs, and then from lungs, the oxygen will go to your body tissues, and this is how the respiration takes place. So the body is complex, the breathing or respiratory organs are present. Respiratory organs are present. Right? You must also have heard about another type of respiration, that is aerobic and anaerobic. Let's talk about that now. We have aerobic respiration and then we have anaerobic. We have aerobic respiration. Respiration is further of two kinds, guys. One is aerobic and another is anaerobic. So aerobic is the one that we do. What is aerobic respiration? First of all, aerobic, wherever the word aero comes, that means it's related with oxygen. So in the aerobic respiration, oxygen is used. Here, oxygen is used. CO2 is produced. CO2 is produced. So when you will oxidize a glucose, it will form the CO2 and water. And also it will form the energy. And the energy content in these two, if we compare, aerobic one produces more energy as comparison to the anaerobic one. So this produces more energy. Fine. In the anaerobic one, less or basically no oxygen is used. Basically no oxygen is used here. The oxygen can be present or absent, but the oxygen will not be used. So it's better to write no oxygen. Okay. So here no oxygen is used. So here CO2 may not be formed, but the energy will be less than the aerobic one. Energy produced is less. So what are the examples? First of all, in us, do we do anaerobic respiration? Yes, certainly we do. So anaerobic respiration takes place in our skeletal muscle. In skeletal muscle, whenever you have low oxygen condition or you're running very fast, you get cramps. Why do the cramps are formed? Because whenever the glucose get burned in the absence of oxygen, it will not produce CO2, it will produce lactic acid. Also in our RBC, alcohol is produced. Yes, RBC does not have nucleus, it does not have mitochondria, so it will undergo anaerobic respiration, because in the aerobic respiration you need mitochondria, but in RBC mitochondria is absent. Yes, so this must be a surprise for you, ma'am. RBC, yes, they also undergo anaerobic respiration. So we have RBC here. What are the other example? Endoparasites in our body. Endoparasites like Ascaristina, yes, they also undergo anaerobic and they also produce alcohol. Yeast fermentation, yeast also produces alcohol. And then we have cellulose fermentation. Cellulose fermentation. All these are the example of anaerobic respiration. So in this, obviously in this chapter, we are talking about humans. So we'll be talking about the aerobic respiration in the entire lecture. So first of all, let's see what are the various respiratory organ in the entire animal kingdom. So guys, wherever I put star, the question can be asked from this topic. Okay, so in the respiratory organs, how does the respiratory organs different in different organism? For example, right now, wherever you're sitting, there must be some ants going on, right? Or ants are running here and there, right? So ants are insects, so they have different kind of respiratory organ. We are different, we have lungs. If you'll see in the water, organisms have different types of respiratory organ. So why different types of animals present on the same planet have different types of respiratory organ? The reason is, first of all, their habitat. You are living on land, whereas fishes, they are living in water. So basically, if their habitat is different, the mode of respiration will be different. Second, the complexity. Ant is a simple organism comparison to us. So according to the complexity of organism, they have different types of respiratory system. Like in sponges. In sponges, the complete organization is cellular level. No complexity at all. So they respire through simple diffusion. And what about us? We have lungs. We are quite complex. We are mammals. So we respire through lungs. So respiratory organ, the types of respiratory organ depends upon first the habitat of organism and body complexity of organism. Body complexity of organism. Let me show you with the examples. And guys, this is really important because, you know, this is a kind of a topic that falls into two categories of chapters. One is animal kingdom, another is breathing and exchange of gases. Then why to leave it? All right, so let's get started with this one. In the flatworms, in the poriferans and ciliate, you can see all these organisms, they're quite simple. They have a, you know, cellular level of organization, they have tissue level of organization, and these flatworms, since they are parasites, and organ level of organization is still there, but they are parasites, so body is quite different, right? So all these will respire through simple diffusion over their entire body surface. For example, if this is a poriferan, right? So here oxygen can enter and CO2 can leave directly through the body. Just like that, here CO2 will leave and oxygen will enter. Similarly with the ciliate. So all these organisms, what? Flatworms, poriferans and ciliate, they respire through simple diffusion. Fine. Next. Earthworm. Earthworm, if you have ever seen earthworm, I don't know what it is said in your language. Let me know in the comments from where do you belong and in what language do you say earthworm. For example, I am a Punjabi. So in Punjab, we call it as Gandoa, right? So earthworm, if you'll see anywhere in the soil, it usually comes out during rainy season from the soil, and they are very slimy. They have a slimy body and it's very shiny, right? It's just like it has a mucus layer on it. So its skin is quite moist. That means it has more water content. Fine. So it has very moist cuticle or skin. So this type of respiration can directly take place by the skin. So you'll not call it simple diffusion. Yes, simple diffusion is occurring here, but skin is the mode of respiration because skin had blood vessels, and from the skin, the oxygen will go to the blood vessel, then to the tissues. How it is different from this one? Here, diffusion takes place, and oxygen directly got diffused into the body tissues. In this one, from skin, the diffusion will take place, and then the oxygen will enter into its blood vessel, and from blood vessel it will then go to the tissues. Okay, this is how it is different. So here, the type of respiration that takes place through moist cuticle or skin, you call it as cutaneous respiration. So in earthworm, what type of a respiration we have? Cutaneous respiration that takes place through the skin. Fine. Next one. Then we have tracheal tubes. The trachea, these are small tube-like structure. They are innervating the body tissues, and they have these small openings known as spiracle. So spiracles are opening through which the oxygen enters, and through these tubes it goes to the tissues. And this type of arrangement, you can see it in the insect. So in insect, the best example we have is cockroach. So we have already studied the cockroach, guys. So in cockroach, we have this type of system. Then we have gills. So gills are seen in aquatic animals. So in these type of gills, which are not that developed, they can be seen in aquatic arthropod and mollusk. See, insect belongs to the phylum Arthropoda, but insects are terrestrial. But there are certain aquatic arthropods. Let me give you an example, like prawn. They respire through the gills. Fine. Also mollusks, they respire through gills. Then we have the respiratory organ in vertebrates. Earlier, all those organisms, they were invertebrates. They are the vertebrates one. So first of all, we have gills in the fishes. These are the highly developed organs known as gills. So these are gills epithelium. From here, the oxygen can enter because gills take up the water, and water have a lot of oxygen. So from gill epithelium, the oxygen can easily diffuse into it, right? So for fishes, we have gills. Then we have lungs. Lungs in amphibians, reptile, avian, and mammalian. So if you can see here, what's the course of evolution, what you say? These are the less developed, more developed, quite different, highly developed, right? So similarly, in amphibians can respire through lungs. Guys, they can also respire through skin, and the respiration that takes place through skin is cutaneous. Respiration that takes place through gills is branchial respiration. That takes place through skin is your which respiration? That is your cutaneous. The one through gills, branchial, and the one through lungs, okay, the one through lungs is pulmonary. What do you call it as? Pulmonary. Okay. So amphibians are special. They can respire through lungs, through skin, and through buccopharyngeal cavity. That's their buccal cavity. Okay. It also have a very moist skin. And last, gills. So gills are seen in aquatic amphibians and the larvae, which are tadpole. Fine. Then we have reptiles. In reptiles, we respire through lungs. In aves means birds. We also reptile through lungs, but they have additional these sacs known as air sacs. The function of air sac is to get more air inside the lungs and supplement the respiration, to increase the respiration. See, if we also get some of these additional chambers, we can get more air. Just like that, nature has given them the air sac to get more air. Two benefits. One, they will get lighter and they can easily fly. Second, in flying, they need more energy. For more energy, we need more respiration, because when we'll break down the glucose, only then we'll get the energy. So that's why these air sacs are present in the avian lungs, and then we have mammalian lungs. So these are the various types of respiratory organs in the different types of animals. Let's talk about the human respiratory system. So the entire human respiratory system is divided into two parts. One is the respiratory passages, another are respiratory organ. And what is our respiratory organ? Lungs. We have one pair of lungs. If we talk about the respiratory passages, these are the small passages from where your air enters and goes towards the lung. So let's talk about them in the detail. So if I talk about lungs, where it is present, this is a chamber. It's a thoracic chamber or a thoracic cavity. Thoracic cavity. And this is abdominal cavity or abdomen. Thoracic cavity and abdominal cavity, how you demarcate it? You demarcate it with the help of this flap-like structure known as diaphragm. So diaphragm is a muscular flap, guys. Diaphragm is a muscular flap. It's a muscular flap. And this muscular flap helps in the demarcation or dividing this cavity, the thoracic and the abdominal. In the thoracic cavity, you can find these two pair of lungs, like this, two pairs of lungs. So what are these? Lungs. This is one lung, this is the another lung. Fine. So this is the exact location of your lungs. Let's talk about them in more detail. So we'll start with the respiratory passages first, because we'll go through the root of the air. So what do you think? From where do you respire? In the, when you are very little, when you were like preschool kid, at home usually our mother and our father or our brother sister, they teach us this is nose, this is eye, this is ear. Later when you grow up more, they started teaching you from where do you breathe? You breathe through nose. This is what your teacher teaches you, right, in school. So we get to know that we breathe through nose. Okay. So the air enters through these small openings. What do you call them as? Nostrils or nares. What do you call them as? Nostrils or nares. Might a lot of you must or do not know about these in English. No worries. When I was at your age, even I got to know for the first time that these openings are known as nostrils in English, because I belong to that area where we do not get to have a lot of exposure for the English language. So you must be surprised, ma'am, but you speak really fluently and you're teaching in PW English. That's the hard work I did on myself in those years. I watched a lot of English movies and documentaries so that I could increase my skills. So you can also do the same. Nothing is impossible in life. You don't need you do not need the external factors. Sometimes your internal factors can also lead to a lot of determination, and that's enough for you to learn anything. I always have determination to learn new things, like eating with the chopsticks. So, what I used to do, I used to buy noodles of just rupees 20, and I bought two chopsticks at a very cheaper price and used to practice at home. So this is how you learn things. You should have your inner ability and determination to learn things. Anything is possible. Anything is possible. Your other factors are not the parts of determination when the determination comes from the inside, right? Okay. So these small openings, they are known as external nostrils. External nostrils or nares. So the entire this external nostril lead to the inside of the nose, and you call it as nasal cavity. Nasal cavity is divided into two nasal chambers. Inside that we have a bone. So if you have ever touched your nose from inside, you feel a wall in the center, and that is a nasal septum. That's a nasal septum. It is also made up of cartilage. What it is made up of? Cartilage. So what we have external nares. External nares. External nares lead to nasal cavity, and nasal cavity is divided into two chambers, two nasal chambers, left and the right. Left and right. Okay, all right. So the entire nasal chamber is divided into three portions. There is first portion where you can also see small hair. And in the old age, these hairs are very much longer, and you can see in your grandfather and mothers these hairs hanging out from the nose, right? And they also have certain glands known as mucus glands. Which glands? Mucus gland. So whenever you get your nasal cavity inflamed during cold, a lot of mucus comes out from this. Fine. So here we have hair, and here we have mucus gland. You all know what is the meaning of mucus gland? It secretes mucus. So this region is known as vestibular region. So the first region is vestibular region. What is the function of vestibular region? To trap the dust particle. The work of hair and mucus is they will clean your air. They will trap the dust particle. Trap the dust particle. Okay. Then we have this region. This is a respiratory region. The function of respiratory region is to humidify the air. humidify and condition the air. How does it will do? Now the air enters here. So we have done two regions. One is a vestibular region, another is a respiratory region. What is present in the respiratory region? There are present three pairs of bones, 1, 2 and 3. So three bones here, three bones on the other side. Okay. So these three bones are known as nasal conchae. Nasal conchae. So nasal conchae are what? These are bones. They are three pairs. Fine. Total three pairs, three here, three there. So these bones are present in a zigzag manner like this. Just like we have done in the digestion, villi is present. So villi is also kind of a zigzag. Wherever anything is present in a zigzag manner, that means it is increasing the surface area. Why do we want to increase the surface area? Because see guys, if the bone is present in this manner, on the bone will be present skin. That means you have more skin. More skin you have. Now air will have more skin to cool itself. You know what happened? Even if, for example, if you are present in a very cold area, the air is at low temperature, the molecules will have less kinetic energy. We want molecules to be very, moving faster, because more they have temperature, the molecules move faster, more will be the diffusion. Okay, for example, if I have warm air and if I have cold air, the warm air will diffuse faster because it has high temperature, the molecules have more kinetic energy, they are more active. Fine. So for example, if you are living in colder area, the air will enter and touch towards the skin of these nasal concave. Nasal concave bones, they have skin on them. Now this skin will maintain the temperature, give a lot of humid, make a humid environment there, and condition the air. So whenever I'm talking about conditioning the air, that means that skin will increase the temperature of air. It will increase the temperature of air. Okay, and that's important. Why? Because more the temperature, more the diffusion. So they're present in zigzag manner. Present in zigzag manner. Why? To increase surface area. Why do we want to increase surface area? More the area, more air will touch to the skin, and there will be more temperature or more heat will be given to the air. Fine. And this area is the olfactory area or olfactory region. Why do we need olfactory region? Very important, because without that your food will be tasteless and you will not be able to smell your perfumes, right? So here we have epithelium known as olfactory epithelium. So the main function of this olfactory epithelium is olfaction. And what do you call olfaction? Smell. Fine. So now this your external layer lead to the nasal cavity, divided into two nasal chamber. Now nasal chamber will lead to the pharynx, but again in between them will be present internal layers. What will be present? Internal layers. So they will be opening after this, and this opening will lead to the pharynx that we have done in the digestion. Pharynx is a common chamber for food and air, which will lead to the larynx, and larynx to the trachea. Let's see how does it happen. So you have seen this is the larynx. If this is the larynx, larynx lead to a pipe known as trachea. Trachea is covered by incomplete cartilaginous ring or C-shaped cartilaginous rings known as tracheal rings. They are 16 to 20 in number and made up of hyaline cartilage. Which cartilage? Hyaline. So the entire length of trachea is 12 centimeter. This is trachea, right? And what are these? These are tracheal rings. Tracheal rings. How many are there? 16 to 20 in number. And what are they made up of? They are made up of hyaline cartilage. They are made up of hyaline cartilage. Fine guys. Now, what is the purpose of them? They are C-shaped. They are incomplete dorsally. That means if this is the ventral side, this is the dorsal side, they will be present like this. So for example, this is trachea, this is the ventral side, this is the dorsal side, and they are present like this. Do I have ring on the dorsal side? No. So I will say they are C-shape and they are incomplete dorsally. If it would be like this, it would be then complete, right? So we say they are incomplete dorsally. They are incomplete dorsally. They are C-shape. They are C shape. What's the function? They prevent collapse because air is entering and leaving. That may lead to the collapse of the trachea. So to prevent the collapse, it will give a firm, firm like nature to the trachea, and trachea will not collapse at all. This trachea at the fifth thoracic vertebrae will divide into bronchi. Imagine this is a place where your fifth thoracic vertebra are present. What is a thoracic vertebra, guys? Have you heard of vertebral column? Your backbone? The vertebral column, vertebral column is made up of small bones, and these bones, they are known as vertebrae. Your entire backbone is made up of small bones. So the bones, these bones are known as vertebrae, and the one which are present in the thorax region, you call it as thoracic vertebrae. Fine. So where your fifth thoracic vertebra is present, the trachea bifurcate into two, and you call it as primary bronchi. What do you call it as? Primary bronchi. So this primary bronchi will further divide into secondary bronchi. What it will divide into? Secondary bronchi. Just like that, it will form a respiratory tree. A tree with a lot of branches. So they will keep on branching and they will form other structure. Like secondary bronchi will open into tertiary bronchi, and tertiary bronchi will open into initial bronchioles. Right? So we say that your tracheal rings are present right from trachea up to initial bronchiole. So remember, guys, your these tracheal rings are not just present in the trachea. We assume they're present in the trachea only, but no, the tracheal rings are present from trachea up to initial bronchioles. Initial bronchioles. All right. Okay. So now what will happen next? The respiratory tree will keep on branching. After initial bronchioles, what will be formed? After initial bronchioles, terminal bronchioles will be formed, and then respiratory bronchiole. Okay. I'll continue here only. After initial bronchiole, what will be formed? Terminal bronchiole. After terminal bronchiole will come the respiratory bronchiole. After respiratory bronchiole will come alveolar duct. After alveolar duct will be present atria. After atria will be present alveoli or alveolar sacs. Right? So this is the entire route. So you can see in this diagram, you can divide the entire respiratory tree into two portions. One is a conducting zone, another is a respiratory zone. Your respiratory zone starts from respiratory bronchioles and it ends to alveoli. Why do you call it as respiratory zone? Because this is a place where actually exchange of gases takes place. So here exchange of gases takes place. Whereas the conducting zone, it starts from your nose, it ends up to terminal bronchiole. So it starts from nose to terminal bronchiole. So its only function is to conduct the air. That means it's a passage for passing of the air. Air is coming from this one. So the only thing that it can do is it can condition the air. It can humidify the air, it can clean the air, it can warm the air. Fine, only these things can be done by the conducting zone. So what does conducting zone do? Warm, humidify and clean the air. But it will not lead to the exchange of gases. This is a portion where no exchange of gases takes place. You call it as anatomic dead space. So you also call it as, guys, anatomic dead space. That means here no exchange of gases takes place. No exchange of gases takes place. Fine, that's a quite difference. But in this is a question that was asked in the exam on Sunday. So which portion leads to they gave all the functions of the conducting zone, and they asked which of the following function was not performed by the conducting zone. Then you will say the exchange of gases. Okay, all right, or the diffusion of gases. Then we have lungs. So all these respiratory tree, they are present in the structure. For example, if I say this is a trachea divided into primary bronchi. Now this primary bronchi enter into these structures known as lungs. Known as lungs. Okay, all right. So the entire respiratory tree is present in your, yes, inside the lungs. So this is the right lung. As you can see, right lung is broader and shorter, left lung is longer and a little thinner, right? So this one is your left lung and this one is your right lung. So the right lung is a little broader and heavier and little upside. Why? Because here you have liver. So due to presence of the liver here, right lung is little on the upper side. And this one is longer and it has this structure known as cardiac notch in it. What is this cardiac notch? While you were an embryo and you were developing, the heart starts to accommodate and side by side lungs were developing, and we say that heart is present little on the left side. So due to presence of the heart, the lung shape gets deformity, or the lungs gets a little change in the shape. It's not a deformity. Deformity is a wrong word to say here. So because of presence of the heart here, the little shape is formed on the left lung. You call it as cardiac notch. And the center space between the two lungs where heart is present, you call this space as mediastinum space. Mediastinum space. Because the left lung is little thinner and smaller than the right one, so it is divided into two lobes only. How many lobes? Two lobes. This is the oblique fissure. Fissure is anything that divides something externally. So due to presence of this fissure, your lung is divided into two lobes. There is upper lobe, you call it as superior lobe, and then you have inferior lobe. The entire lung is divided into, left lung is divided into two portions, inferior and superior lobe, due to presence of the fissure. Now we have this right lung which is divided into two portions. So we have two fissures here. One, we have horizontal fissure, and then we have oblique fissure. As a result, the lung is divided into two portions, superior lobe, the middle lobe, and inferior lobe. All right. Now, the space present inside the lung where where your air enters, you call that space as pulmonary cavity. Right? So for example, you have the alveoli structures in them. We'll be talking about that in detail. So these alveoli have a space in them that is a pulmonary cavity. So the space present, so there are different kinds of cavities, guys. Okay, so there are three cavities in your lung. One is your thoracic cavity that we have already talked about. Another is pulmonary cavity that is present inside lungs, in the lungs. And the last we have is a pleural cavity. Now what is this? So your lungs, they are protected by double layer bag-like structure. You call it as pleura. So your entire lung is having this double membrane bag-like structure on it. So I'm drawing on the one lung, it's present in the both. Okay. Okay, just let me draw here also, otherwise we'll get confused. Okay, so here we have double membrane bag-like structure. This is single membrane. This is the second membrane. Like this. Okay, a double membrane bag-like structure. You call it as pleura. What do you call it as? Pleura. So pleura is made up of two layers. It's basically a peritoneum. This is what? Pleura. So pleura is made up of two layers. It is made up of two layers. The pink one is visceral layer, and this one, the green one is a visceral layer. All right, green one is a visceral layer because it is present near the visceral organ, and this one is a parietal layer. And together these layers, they are collectively forming the pleura, the double bag-like structure, double membrane bag-like structure. What's the function of the pleura? Inside the pleura is present a space, you call it as pleural cavity. What is this, guys? Pleural cavity. And pleural cavity have a fluid. You heard it right, it have a fluid. And this is pleural fluid. This pleural fluid performs a very important function. First of all, shock absorption. And second, it reduces friction. So in a way, it is providing protection. So the cavity inside that is a pleural cavity. So the cavity inside the lung is a pulmonary, this is pleural, and the one is where lungs are present is a thoracic cavity. Fine. Okay, let's talk about alveoli now. Alveoli or alveolar sacs. Alveoli or alveolar sacs, they are total 300 million in us. Yes, 300 million. So much surface area. So there is more surface area for exchange of gases. For what? Exchange of gases. They are highly vascular. They are highly vascular structure. Highly vascular means you will find a lot of blood vessels. You will find number of blood vessels, a lot of blood vessels. Okay, let's see this one. So if this is the alveoli, this is the alveoli. This alveoli have the epithelium known as simple squamous epithelium. What is this epithelium? Simple squamous epithelium. So air sacs or the alveoli, they have which epithelium? Simple squamous. So it has certain specialized cells in them. So the first type of these cells are simple squamous. Okay, and this is the lumen of alveoli. Now apart from that, they also have certain different cells. So you can find certain cells here. One such cells are these type 2 alveolar cell, and then we have cells known as dust cells, which are macrophages. So these are dust cell or macrophages. All right, and then we have these pink color cell. What are these? These are type 2 alveolar cell. These are very important. These cells, they secrete a phospholipid. So a question of was also there in this exam of NEET exam. Lecithin. Lecithin is a phospholipid. Though it was a question from biomolecules, but it also have an application here. So it secretes a phospholipid, which is lecithin. The function of the lecithin is to decrease the surface tension. It decreases surface tension. This lecithin decreases surface tension and prevents the collapse. Prevent collapse. You know, so for example, in balloon, if you fill the air in the balloon, it will expand. But if you take out the air from the balloon, it will collapse. So we have to give something to the balloons that they will not collapse. And this is what your lecithin and tracheal rings it does. Okay, so tracheal rings prevents the collapse of trachea and the bronchioles, whereas here lecithin, because they do not have any bronchiole, those tracheal rings they end up to initial bronchiole. Later on, anything does not have the these tracheal rings. So that's why we need to have something that would prevent its collapse, and that is the lecithin secreted by type 2 alveolar cells. Fine, guys. So let's talk about what are the various bones present around the lungs. So lungs, they are also protected by rib cage. They are also protected by rib cage. Now rib cage is formed by three types of bones. One are your thoracic vertebrae. Second are your ribs, and third is sternum. Okay, how it is present? For example, ventrally, sternum is present, and dorsally, thoracic vertebra are present. Okay, and ribs are present like this laterally. So this entire structure is your rib cage. So this is the sternum, these are the ribs, and this is the thoracic vertebrae. So we say that this is the dorsal side. They are present dorsally, ventral is present, the sternum is present ventrally, and ribs are present laterally. Okay, let's solve the question now. All statements are correct except during swallowing, glottis can be covered by a thin elastic cartilaginous flap called epiglottis. Pharynx, the portion of which is the common passage for food and air. The lungs are situated in the thoracic chamber, which is an anatomically an airtight chamber. Rib cage is formed by sternum and lumbar vertebra. So in the previous class, we have done that epiglottis is a cartilaginous flap-like structure, and that belongs to which part? Larynx. So that's why I have not repeated in this class, because we have already done in the digestion part. Okay. So this is a larynx, guys. Yes. If you can see here, this is the larynx. This is the larynx, your voice box. So this larynx, it has an opening known as glottis. And this glottis is covered by a flap-like structure, elastic flap-like structure known as epiglottis. So this is true. Whenever you eat food, this epiglottis closes the glottis so that the food does not enter into your trachea, which is also known as windpipe. Okay? Alright. So if it accidentally does while you are talking and eating, you get cough. Yes, pharynx is a common chamber for food and air. True. Yes, it is present in an airtight chamber. Can you see any air coming out from this one? No. So thoracic cavity is completely an airtight chamber. Whereas rib cage is not formed from lumbar, rather thoracic vertebrae. Okay. So answer to this question will be 4. Next. Trachea is a straight tube extended up to the mid-thoracic cavity, which divides at the level of, where does that it divides? It divides at 5th thoracic vertebra. 1. Next. The inner pleural membrane is in contact with lung surface, body wall, alveoli, and the bronchi. What is the inner pleural membrane? Inner means they are talking about the visceral one. Outer means they are talking about the pleural one. So it is in the contact with the lung surface. Right, guys? So the one which we have done here, the visceral layer. The visceral layer is also, because it's present on the inside, so you call it as an inner pleural membrane. And parietal one is present outside towards the body wall, so it is the parietal one. Okay, all right. So next question, match the following. Gills. So where do you find gills? You have to match it. Gills are present in the fish. Lungs. Lungs are present in the aves. Okay. Simple diffusion by body surface in the flatworms and cutaneous in the earthworm. Simple, right? Okay. Next one. Dash have a network of tubes to transport atmospheric air within the body. Amphibia, insects, aves, and reptiles. So we are talking about tracheal tubes or trachea, and that's a feature of your cockroach. And cockroach is an insect. Answer is 2. Amphibians respire through lungs, gills, buccal cavity, and even the skin, whereas the aves through lungs, reptiles through lungs. Aves have a special air sacs in their lungs. Next we have is respiration involving the following step. So now we are going to talk about our respiration. So what are the various steps in our respiration, or how does your glucose is breaking down into CO2 and water and giving you energy? So we have to start it from the start. So first of all, what will happen? You will take in the oxygen and throw out the CO2. So first step is ventilation or breathing. So what happens in pulmonary breathing or ventilation? By which atmospheric air is drawn in and CO2 rich alveolar air is released out. Next, diffusion of gases, oxygen and CO2 across the alveolar membrane. Now alveoli will take the oxygen and give it to the blood, because I've said alveoli is highly vascular, and you call it as exchange of gases. And then there will be transport of gases from blood. The entire gases will go to the tissues, because blood helps in the transport of gases. Then diffusion will take place between blood and tissue. So we say that exchange is taking place at 2 places. Exchange of gases takes place at 2 places. One between blood and tissues, another between blood and alveolar membrane. Here also exchange. Okay. Next, then the cells will pick up the oxygen and give you energy. This is what entire chapter contains, and this is what we will start to study now. Okay, all right. So first of all, we'll talk about breathing. So entire breathing or pulmonary ventilation takes place in 2 steps. One is you inhale the air, another you exhale the air. So inspiration is also known as inhalation. What do you do in inhalation? We take in the fresh air. And in exhalation or expiration, what do? We take out or exhale the bad or foul air. Fine. So how does air moves? Like blood moves from area of high pressure to the low pressure, just like that, air also moves from area of high pressure to the area of low pressure. For example, if I have in this area high partial pressure of oxygen, it will move from high pressure to the low pressure, right? If I have more partial pressure of air to the outside and less in my body, the air will move in. First thing is this. So air moves from area of high pressure to the area of low pressure. Now, whenever there is increase in pressure, there will be decrease in volume. This is what we say, Boyle's law. These two fundamentals are very much important in inhalation and exhalation. Yes, that's true. Let's see how. So let's talk about it in detail. So what happened? We say that the movement of air in and out of the lung is carried out by creating a pressure gradient between the lungs and atmosphere. So if you want to move the lung, you need to create a pressure gradient. What is a pressure gradient? What is a pressure gradient? Difference in partial pressure or the pressures. Difference in pressure. For example, if I say these are the lungs and I want lungs to take the air. If I want inspiration to occur, if I want to take in the air, I will make sure my pressure inside the lung should be less so that the atmospheric pressure is more and air will move in. How can I decrease the pressure? I can decrease the pressure if I increase the volume. So I will, with my efforts, I will increase the volume and the pressure will decrease, air will move in. So what I will do in exhalation? In exhalation, what I will do? I will create more pressure inside the lung. How will I create more pressure inside the lung? I will, with my efforts, I will decrease the volume and increase the pressure. Now, atmospheric pressure is low and air will move out. This is what we will do in breathing. So, how are we increasing and decreasing the pressure? We have to make effort, and efforts in our body is made by our muscles. So yes, muscles are involved in inspiration and expiration. Now what are these muscles? Let's see. So muscles of breathing. This is very important. From here the question is asked. First question can be asked from this portion about the volumes. For example, they ask during inhalation the pressure inside decreases. True or false? True. During exhalation, the pressure inside the lungs increases. True or false? True. Second question, the pulmonary volume during inhalation increases. This is also true. The pulmonary volume during exhalation, it decreases. This is also true. First type of a question can be asked from this portion. Second type of a question can be from a muscle portion. Now, what are the muscles? So, imagine this is your rib cage. In the rib cage, we made sternum, we made thoracic vertebrae, and we made ribs. This is what it was. Yes. So there are muscles present in between the ribs, and the space in between the ribs is intercostal space. The space in between two ribs is known as intercostal space. This space is intercostal space. What is intercostal space? The space between ribs. Okay. Okay, let me label this also for you. This is ribs, this is sternum, and this is what it is? Yes, thoracic vertebrae. Now, there are muscles present in between the ribs. The name of the muscles is on the basis of how they are positioned. Some are present towards inner side, they are internal intercostal muscles. Some are present towards outside, they are known as external intercostal muscle. So here we have a muscle, guys, listen to me. This is, for example, external intercostal muscle. And this is a muscle which is internal intercostal muscle. Then we have here diaphragm. What do we have here? Diaphragm. And diaphragm is also a muscular flap-like structure. Diaphragm is also a muscular flap-like structure. So the diaphragm muscles, they are also important in inhalation. So diaphragm or phrenic muscle. All right. Diaphragm and phrenic muscle and external intercostal muscle, they both are responsible for normal inhalation or normal inspiration. Whereas your intercostal muscle and there are certain abdominal muscle. And which muscle? Abdominal muscle. Okay, I will not mark it here, otherwise you'll get confused. Both these muscles are responsible for your forceful exhalation. So this muscle is for, okay, forceful exhalation. Ma'am, forceful? Do we do forceful exhalation? Do it. This is forceful. Right now I'm breathing. That's normal. Right? So yes, we also do forceful exhalation. And there are certain abdominal muscles present here. These are abdominal muscles. They are also responsible for forceful exhalation. Just put an effort and throw out the air. You will yourself feel your abdominal muscles are stretched. Right? So forceful exhalation. All right. So these are various muscles. Let's talk about the physiology part now. So what happened during inspiration? So whenever you are inhaling, you inhale by two methods. One, you inhale normally, and second, you inhale forcefully. You inhale forcefully. So whenever we are inhaling normally, we are contracting two muscles. One is external intercostal muscle. Right? Whenever, for example, right now you are inhaling, you are contracting your external intercostal muscle and your diaphragm muscle. So what will happen if your external intercostal muscle contract? When the external intercostal muscle contract, your entire rib cage will move upward and outward. You just do it with the forceful also. You'll see your chest coming out. So what this external intercostal muscle does, it will raise the sternum, okay, or entire rib cage. So rib cage will move upward and outward, upward and outward. As a result, towards dorsolateral axis, like I'm doing like this, okay. So I am raising my chest or my rib cage. As a result, on the dorsolateral axis, the volume will increase. Okay. So what will happen when they will contract? They will move rib cage upward and outward. As a result, what will happen? The volume increases. At which axis, guys? Dorsoventral axis. At this axis, your volume will increase and the pressure will decrease. And when the pressure decreases, air moves in. All right, this is what we do. And the volume and the pressure, it changes in all the three cavities: thoracic, pleural and pulmonary. Because first of all, the chest will expand, thoracic cavity will expand, then pleural cavity will expand, then pulmonary cavity will expand. And all the volumes are increased in all these three cavities. Fine. And during contraction of the diaphragm, what will happen here? When they will contract, they will, for example, if this is your thoracic cavity, okay, this is thoracic cavity, this is a diaphragm. So when it will contract, what will happen? It will be pulled downward like this. So now you can see here the volume of thoracic cavity and here the volume of thoracic cavity. What do you think? Where is the more volume? When the diaphragm contracts. So when the diaphragm contracts, the diaphragm flattens. And when the diaphragm flattens, there will be increase in volume in anterior posterior. This is anterior and this is posterior position. Always your head is on the anterior position and your foot are on the posterior. So it will increase the volume in anterior posterior axis. And when this increases, what will happen? The pressure will decrease and air moves in. Air moves in. Fine. What about forceful? In the forceful one, what happened in the forceful kind of inspiration? Your another muscles contract. These muscles are present toward this portion. What are these named? Scalene muscle, pectoralis, and sternocleidomastoid. All these are muscles which are present around your rib cage. Sternocleidomastoid. All these are present around your rib cage. So when these muscles contract, they will move the rib cage more upward and outward. And outward. Right? The rib cage will move more upward and outward. Fine, guys. So that's about your inspiration. Let's talk about the expiration. What happened in expiration? Again, expiration is of two types. One is your normal, like you're doing right now, and one is forceful. So what happened in this one? So whenever you are exhaling normally, you are not putting any effort, whatever muscles of your inhalation have been contracted, whether they are of the forceful or they are of the normal inhalation, they will relax. And when they relax, the entire chamber and the volume of the chamber or rib cage, it will move back to its original position. As a result, the volume decreases and pressure increases, right? Whatever they have done, they will come back to their original position. So what happened here? During normal, all muscles relax, right? As a result, there will be decrease in volume, increase in pressure. Air moves out. What about the forceful? In forceful, your internal intercostal muscle and abdominal muscles contract. So what they will do, when it will contract, it will do opposite to the external. External was putting your rib cage upward and outward. It will put inward and downward, right? Rib cage moves inward and downward. So when abdominal muscles contract, for example, the abdominal muscle contract, they will push the diaphragm to the upward, right? They will push diaphragm upward. What will happen? The volume decreases, pressure increases. Same here, volume decreases, pressure increases, air moves out. Air moves out. Very simple. Good to go, right? So this is how you breathe, guys. So questions are generally asked from which muscle will contract during which mechanism, inhalation or exhalation, and what will happen to the volumes and the pressures. Fine? Okay. Moving further, on an average, a healthy human breathes around 12 to 16 times per minute. The volume of air involved in breathing movements can be estimated by using a spirometer, which helps in clinical assessment of pulmonary function. So we say that how many times do we breathe in a minute? 12 to 16 times, right? And can we measure the breath, the volumes? Definitely we can, and we can do it with the help of this machine. You call it a spirometer, and spirometer was very much important in or during the time of COVID. Yes, it was really important. Spirometers is inbuilt in the ICU machines, ventilator machines, which can check what is your pulmonary volumes and capacities. Now, what are these pulmonary volumes? The amount of air you take in and take out, and that's a pulmonary volumes and capacity. Now, what are these pulmonary volumes? Volumes and capacity. Capacity is always the sum of volumes. It's the sum of volume. So it's just about the quantities, how much air you're taking in and taking out. We are talking about numbers now. First of all, guys, we have a volume known as a tidal volume. What does tidal volume says? Tidal volume says it's the amount of air you take in and take out normally when you're not putting any effort. Right now, you're doing that same. You're just normally inhaling 500 ml of air and you're also exhaling 500 ml of air. That's it, right? So what is the tidal volume? The amount of air in or out normally. Its volume is, its quantity is around 500 ml. Fine. This is really important. The questions are asked from this portion. Now, second, what if I put some effort and I do forceful inspiration? So during forceful inspiration, I am taking, for example, I did forceful inhalation. Like, you also do it with me. So this time guys, you are taking 500 ml tidal volume inside your lungs and you are also taking some extra amount of air. And that extra amount of air or additional amount of air is your inspiratory reserve volume. So what is inspiratory reserve volume? It's the additional or extra amount of air taken in during forceful inhalation. During forceful inhalation. And what's its quantity? 2500 ml to 3000 ml. Now I said during forceful inhalation, I am taking tidal volume also and inspiratory reserve volume also. So what total amount I am taking? I have to sum up these two and you call it as inspiratory capacity. So if I do the total of these two, it will become inspiratory capacity. Inspiratory capacity is the total volume of air I am taking in during forceful inhalation. Extra or additional amount of air taking in during forceful inhalation is IRV, inspiratory reserve volume. And what is IC? IC, inspiratory capacity, is tidal volume plus IRV. What does it say? Total volume of air taken in during forceful inhalation. During forceful inhalation. Fine. Now what about expiration? We also do forceful expiration, isn't it? So that is expiratory reserve volume. Just like during forceful inhalation, you take TV and you take IRV. So when I am exhaling forcefully, I am also exhaling TV, the tidal volume, and I'm also exhaling some extra amount. That extra amount I am exhaling apart from TV is ERV, expiratory reserve volume. What is expiratory reserve volume? It's the extra or additional volume of air exhaled during forceful exhalation. During forceful exhalation. Its amount is from 1000 to 1100 ml. Right? So what will be EC? Expiratory capacity, guys? What will be EC or expiratory capacity? Expiratory capacity will be then tidal volume plus ERV. Okay, so what does it say? Total amount of air, just like that. Total volume of air taken in during forceful inhalation. And this will be total volume of air taken out during forceful exhalation. Right? This is the capacity. Now let's talk about one more volume. We have tidal volume, we have inspiratory reserve volume, we have expiratory reserve volume. One more volume we have, and that is residual volume. So all volumes I'm writing it in the green one. So what is residual volume? Whenever you exhale forcefully, whenever you exhale out forcefully, at that time some amount of air remains in the lung. Why does it remains in the lung? Because you always have negative pressure inside the lung. A little negative pressure is always maintained so that some amount of air always remain inside the lung, and that's residual volume. But why we are doing it? To prevent the collapse, to prevent the collapse of the alveoli, right? So residual volume is the amount of air remains in the lung even after forceful exhalation, even after forceful exhalation. Okay, for example, if this is your lung, so we say some amount of air always remain in the lung even if you have taken out the ERV and even if after you have taken out the tidal volume. So whenever you're doing forceful exhalation, what goes out of the lung? ERV and tidal volume. What will be remained? That amount is residual volume. Okay. Now one capacity, functional residual capacity. Okay. What's the value of this one? 1100 to 1200 ml. Okay. Then we have functional residual capacity. What is this? So when you were exhaling forcefully, the air that remains in the lung was your residual volume. What if you do not take out ERV, you only take TV? If I'm saying the person is only taking out TV, not ERV, that means the person is exhaling normally. This is what I say now. If I say there is a person who is not taking out the ERV from the lungs, but only TV from the lungs. How the person is exhaling? Normally. So, amount of air that remains in the lungs after normal exhalation, if FRC, functional residual capacity. What is functional residual capacity? Total volume of air remains in the lung after normal expiration. Now, what's the difference between the two? What's the difference? Both are the one which remains in the lung. The difference is in the forceful and normal expiration. Okay. Now, one more capacity that we have is vital capacity. What's a vital capacity? So, vital capacity is formed by the addition of these three: TV, inspiratory reserve volume, and expiratory reserve volume. For example, if I say there is a person, there is a person. This person is inhaling forcefully. This person is also exhaling forcefully. So what this person is taking in and taking out? During inhaling forcefully, this is taking tidal volume plus IRV, and during forceful exhalation, it is also taking tidal volume and ERV. What's the common? Tidal volume is common. So we say that vital capacity is formed by the union of three things. Vital capacity is formed by the union of three things. One is IRV, another is tidal volume, and whenever I'm saying tidal volume, I'm taking both in and out. I'm writing both in and out, okay? Plus ERV. Okay, now another type. Another is total lung capacity. What does total lung capacity says? For example, a person inhaled forcefully. Inhale forcefully. When a person inhales forcefully, inside the lungs, person already have residual volume and already have ERV. So while a person is inhaling forcefully, what it will take inside? Tidal volume and IRV. So this, if I total them up, this will become TLC, total lung capacity, which is formed by the union of IRV plus TV plus ERV plus RV. Or I can also write it like that, vital capacity plus RV, because this is vital capacity. Okay. What is vital capacity? Both times you are inhaling and exhaling forcefully. So at that time you will also get IRV, you also get ERV, and you will also get tidal volume. Whereas in the total lung capacity, so whenever you are inhaling forcefully, at that time, what is present inside your lungs is a total lung capacity. So, you know what kind of questions can be asked? This is quite important. First, they can ask you about capacities and what they are made up of. Like TLC is made up of is equal to this. Second, definition based question. Third, the quantity based. Like I have already told you, residual volume is this much. ERV is this much. IRV is this much, and tidal volume is this much. So these volumes, they are also very important. Okay, so let's solve question for more clarity, guys. During inspiration, the volume of, okay, so before starting the question, let me explain you one more topic, and that is pulmonary minute ventilation. Minute ventilation. What is minute ventilation? We say that how much amount of air you have breath, or how much ventilation you have done in 1 minute. How much ventilation or breathing you have done in 1 minute is minute ventilation. And this has a formula, that's the number of breaths per minute into tidal volume. So what is minute ventilation? The amount of air inhale or exhale in 1 minute. Amount of air in or out in 1 minute. So how much breath are there? 12 to 16. Multiply it with 500. What will be the answer? Yes. What will be the answer if you multiply it with the 500? Yes, guys. What will be the answer? Just do it with upper or the lower limit, anyone. It will be approximately 6 liter per minute. Approximately 6 liter per minute, or 6000 ml per minute. Fine. So let's now do the questions. During inspiration, the volume of thoracic cavity increased because of, so it is saying whenever you are inhaling, we say the volume increases. Why does volume increases? Now you have to tell the muscles, okay? Contraction of diaphragm and external intercostal muscle, very true. Okay, next, relaxation of diaphragm. Diaphragm does not relax. Contraction of diaphragm and relaxation of, no. Always contraction of muscle leads to the change in the volumes. Relaxation, no. So answer to this question will be 1. Okay, you can read it. You can pause and then you can read the question nicely. Next, additional volume of air a person can inspire by a forceful inspiration. It is saying the person is forcefully inspiring. At that time, what extra or additional volume of air that person is taking? What do you call it as? You call it as IRV. If it would be expiration, then it would be ERV. Answer is 1. Okay, so this is a one type of a question. Next, read the following statement and find the incorrect one. Conducting part is the site of actual diffusion of oxygen and CO2 between blood and atmosphere. No, that's a part or the function of respiratory portion. Second, the trachea, primary, secondary and tertiary bronchi and initial bronchioles are supported by incomplete cartilaginous ring. Very true, because here from trachea to initial bronchioles we have cartilaginous ring and they are incomplete. They are not complete. The movement of air into and out of the lung is carried out by creating a pressure gradient between the lung and atmosphere. Yes, you have to create a difference in pressure. The volume of air involved in breathing movement can be estimated by using spirometer. This is also true. So we have to find the incorrect one. Answer is 1. Next. Match the following columns. Again, second type of a question on the basis of amount. IRV. Maximum is IRV. ERV is the 1000 to 1100. RV is around 1200 and TV is 500. Okay. Next. A healthy man can inspire or expire approximately dash amount of air per minute. We have just done it now. 6000 to 8000 ml per minute or 6 liter to 8000 liter. If we take upper limit and lower limits, both. How does it come? If I take 12, it will be 6 liter. If I take the amount of 16, then it will be 16 breath, then it will be 8 liter. Answer is 3. Next. Next, we have the very important topic, that is exchange of gases. Exchange of gases. How does the exchange of gases takes place? First of all, the question is, where does the exchange of gases takes place? First, the exchange of gases takes place between alveoli and blood vessel. Second, take place between blood vessel and tissues. So this is the most common question asked. Tell the 2 places where exchange of gases takes place. 1, between alveoli and blood vessel. Second, between blood vessel and the tissues. Okay. All right. Now, what are the factors that will lead to the exchange of gases? Exchange means taking in the oxygen, giving the CO2 and vice versa. There are certain factors which are very much important for the exchange of gases. The first is partial pressure or the difference in the pressure. First is the pressure gradient. So earlier when we were doing breathing, we were talking about pressure of air, right? Now we are talking about gases. Now we will talk about partial pressure. What's the difference? For example, I have this amount of a thing. This is air. So for air, I'll be always using pressure P. But this air is made up of oxygen, CO2 and other gases. So the pressure of individual gas in a mixture of gases is a partial pressure. So if I say this is partial pressure. So what is the partial pressure? Pressure of individual gas. Pressure of individual gas in the mixture of gases. In the mixture of gases. Fine. So first factor that affects, these are factors that affects the exchange of gases. First is the pressure gradient. So we say more the difference in the partial pressure, more the exchange. For example, if I say in air the partial pressure of oxygen is around 159 mm of mercury and in the alveoli it is 104. There is so much difference. Yes, there is so much difference. This is high pressure, this is low pressure. So air can easily move. It can move in. right? If I say in air the pressure, partial pressure of oxygen is 159 mm of mercury, in alveoli it is 104. So here you can see the partial pressure of oxygen is less in alveoli, so from air it can easily move inside. So more the difference, more the difference, more the exchange of gases, exchange of, right? All right, moving further. So the one factor is change in the pressure gradient. Let's talk about the other factor. Second factor is solubility. Second factor is solubility. So if I say one gas have more solubility than the other, so one gas which have more solubility can easily pass through the membranes, and it can easily, you can say, get exchange. So more the solubility, more the solubility, more the exchange. So as we say, CO2 have 20 times more solubility than oxygen. So it can easily be exchanged than oxygen. Fine. Third factor is thickness of the diffusion membrane. Thickness of diffusion membrane. Now, what is this diffusion membrane? Let's see. So we say that if this is the alveoli, this is the simple squamous epithelium of alveoli, and this is the lumen. And as you know, every epithelial cell have connective tissue with them. So this is the simple squamous epithelium of what? Alveoli. Below that will be present a basement membrane, as every epithelium is incomplete without that. So here will be the basement membrane. And then it will be surrounded by blood vessels. So as you also know, the blood vessels or the capillaries, they also have epithelium in them, and that is a simple squamous epithelium, generally known as endothelium. So whenever oxygen has to enter from alveoli lumen to the blood vessel, it has to diffuse through all these three membranes. What are these membranes? First membrane is your simple squamous. Second is the basement membrane. And third is endothelium. What is this endothelium? Endothelium is a simple squamous. Endothelium is a simple squamous of your blood vessel. So, simple squamous of blood vessel. Okay. So these three layers form your diffusion membrane. So more the thickness, more time it will take for the gases to get exchanged. So we say that less the thickness, more the exchange. Less the thickness, more the exchange. Okay. Now let's move further and talk about the actual exchange of gases. So how does exchange of gases takes place? All right, guys. So you must have seen this diagram in NCERT. This is the alveoli and this is your heart. First of all, to understand this, you need to understand the circulatory system. What? Yes, you heard it right. So your heart is four-chambered. Heart is four-chambered. Fine. So in this four-chambered heart, you can see if this is the right side of the heart, this is right atrium, this is the left atrium. This will be the right ventricle, this will be the left ventricle. Fine. So we say that from lungs, listen to me very carefully, okay? So we say that from lungs, the oxygenated blood enters in the left atrium, right? And from left ventricle through aorta, this blood goes to the tissues so that it can give the oxygen, This is what we study, right? Imagine this is the tissues. or these are the tissues. Tissues need oxygen and they will give its carbon dioxide. Why tissues are doing these? Because these are your body tissues, your cells, which are doing oxidation. What are they doing? Oxidation. Fine. So we say that from alveoli, the oxygenated blood, it enters here through pulmonary vein. This is pulmonary vein, and the blood in pulmonary vein is oxygenated. This is an exception. Veins carries deoxygenated blood, but there is one exception here. Pulmonary vein carries the oxygenated blood. Otherwise, veins carries deoxygenated blood. I think I said oxygenated earlier, so pardon me. So we say veins carries deoxygenated blood, but this is an exception. Pulmonary vein carries oxygenated blood, and this is systemic arteries. Systemic arteries, and systemic arteries gives blood to the tissues. Let me put down the arrows for more clearance. So here the blood is moving like this to the atrium, then ventricle, then from here to the tissues. All right, now once this blood had given the oxygen, okay, because it is carrying the oxygen, it will give oxygen to the tissues and pick up the CO2. Once it has picked up the CO2, so the blood will become deoxygenated and it will enter into the systemic veins. Which veins, guys? Systemic veins. Okay, so here we have systemic veins, and we say systemic veins carries deoxygenated blood. So here the deoxygenated blood will now enter into the right atrium. From right atrium, it will enter into the right ventricle. From right ventricle, the pulmonary artery, which artery, guys? Pulmonary artery, will enter into the alveoli so that it can pick up the oxygen, because the blood is deoxygenated. So this is pulmonary artery. This is pulmonary artery. So this is also an exception, that pulmonary arteries carries oxygenated blood, but this is an exception. This one carries deoxygenated blood. This is how circulation is. So let's see how the exchange of gases takes place. Now you got to know this oxygenated blood will give oxygen to the tissues and pick up CO2, and this one will give CO2 to the alveoli and pick up the oxygen. This is how this blood is oxygenated. Fine. So now we say that here all the partial pressures are written. We say that in the atmosphere, the partial pressure of oxygen is 159 mm of mercury, whereas the partial pressure of CO2 is 0.3 mm of mercury. And you all know the exchange of gases takes place based on only one phenomenon. And what is that? The change or the difference in the partial pressure or the pressure gradient. So the pressure gradient will lead to the exchange of gases. Fine. So here we say that, guys, see very carefully, the partial pressure of oxygen, this is alveoli, guys. The partial pressure of oxygen in the alveoli is 104. The units will be same mm of mercury, whereas the partial pressure of CO2 will be 40. Okay. Now, the blood which is coming here, what is that blood? The blood that is coming here is deoxygenated. That is in the pulmonary artery. So we say that deoxygenated blood is present in the pulmonary artery and in the systemic vein. So this blood have the partial pressure. You can see here it has oxygen in the level of 40 and CO2 with the level of 45 mm of mercury. Okay, this is the partial pressure of oxygen and CO2. So this blood is entirely same, have same concentration of oxygen or the same partial pressure of oxygen and CO2. So if I say the partial pressure of oxygen is 40 and partial pressure of CO2 is 45. Let's compare oxygen first. Here oxygen is 40, here oxygen is 104. Now what do you think? How does exchange will take place? Because oxygen is at higher pressure, this is at lower pressure. So this blood have 40 oxygen and alveoli have 104. So now it will lead to the movement of oxygen into the blood. Same in this blood, the partial pressure of CO2 is 45, whereas the partial pressure of CO2 in the alveoli is 40. If I say 45 partial pressure of CO2 is here. Okay, and here it is 40. Now it will lead to the exchange of CO2. CO2 will move in the alveoli. Same will take place between alveoli and the atmosphere. This is in the atmosphere. Okay, in the atmosphere, the partial pressure of CO2 is 0.3 and here the partial pressure of CO2 is 40. High pressure, low pressure, CO2 will move out. Here, partial pressure of oxygen is 159, here 104. here oxygen will move in. Similarly, now the blood has become oxygenated because it has picked up the oxygen. Reason, because partial pressure of oxygen in the deoxygenated blood was 40 and in alveoli it was 104. So oxygen enters here. Now in the pulmonary vein and systemic artery, we have same condition. They have oxygenated blood. So the partial pressure of oxygen, as it is also written there, is 95 mm of mercury, and partial pressure of CO2 is 40 mm of mercury. Okay, now in the systemic arteries, guys, as you can see here, partial pressure is 95 for oxygen and 40 for CO2. Let's talk about tissues. In tissue, the partial pressure of oxygen is 40 and partial pressure of CO2 is 45. Let's compare now. Here you can see, guys, that this is 95 and this is 40. High pressure, low pressure. What do you think? Where should the oxygen will move? It will move inside the tissues. Now here CO2 is 45 and here CO2 is 40. Now where does the CO2 will move? CO2 will move here inside the blood. So this is all just on the basis of difference in the partial pressure, the gases are moving just from high to low. You just need to know the values here. You just need to know the values here and everything will go smoothly, and you need to understand the type of circulation. The gases will always move from high pressure to low pressure. Okay. All right. Let's move further and talk about the transport of gases. So this was about how the gases exchange take place. Like we said, here exchange take place at two positions, one between blood vessel and alveoli, another between tissues and the blood. This is what it happened. But how does this entire transport of oxygen is taking place, or how does the entire transport of CO2 taking place? We have to see that. Okay, so for that you need to understand what are the two important gases. One is the oxygen, and second is the CO2. Second is your CO2. So if I talk about how does they get transported, oxygen, 97% of oxygen is getting transported with hemoglobin. How does with hemoglobin? If you know the structure of hemoglobin, I am writing it in short form, HB. It consists of two parts. One is heme, another is globin. Globin is made up of amino acid, whereas heme part, it contains iron. It contains iron, right? It contains iron. So we say that one hemoglobin contains 4 iron. And one iron binds to one molecule of oxygen. That means O2. I'm not talking about nascent oxygen. O2, right? So here 4 iron can bind to 4 oxygen. So we say in one hemoglobin, 4 oxygen can easily get transported. So when it binds with the hemoglobin, it binds to which part of the hemoglobin, guys? To the heme part, and it leads to the oxidation of iron. When oxygen binds to iron, it undergoes oxidation. So we say that here hemoglobin bind to oxygen in the reversible process. This is the reversible one, and form oxyhemoglobin. So 90% part of 97% part of oxygen transport with the help of binding state with the hemoglobin. What for the rest 3%? The 3% is transported in the dissolved form in plasma. Fine. Okay, so let's talk about CO2 in a later sections. First, let's talk about the oxygen more. Now, if I say if a person is having 15 gram of hemoglobin per 100 ml of blood, this is what we say, that a person who is healthy have around 12 to 15 gram of hemoglobin. In male, it is always around 14 to 15. In female, it is less because female undergo menstruation. So we say that in a healthy person, 15 gram of hemoglobin is present per 100 ml of a blood. Okay. Now, if I say 15 gram of hemoglobin is present in 100 ml of a blood, and 1 gram of hemoglobin can carry around 1.34 ml of oxygen, then how much 15 gram of hemoglobin will carry oxygen? If I say 1 gram of hemoglobin can carry 1.34 ml of oxygen, then 15 gram will carry 15 into 1.34 ml of oxygen, which is approximately 20 ml of oxygen per how much blood? 100 ml of oxygenated blood. Why now I am talking about oxygenated blood? Here I have written only blood, because now I am saying the 15 gram of hemoglobin is completely saturated with oxygen. So that means the blood will be oxygenated. If I say all the molecules of hemoglobin in the blood are having oxygen, so definitely the partial pressure of oxygen is higher. So the blood will be oxygenated. Okay. Now, out of this, guys, listen to me very carefully. Out of this 20 ml, out of this 20 ml, right? Out of this 20 ml, whenever you are doing rest, because you don't need much oxygen, your tissues does not need much oxygen, 5 ml oxygen goes to tissues, and 15 ml oxygen goes to blood. Whereas when you are doing certain exercise, or you are doing something strenuous exercise, or you are working, or you are in active physiological condition, 15 ml of oxygen goes to tissues and 5 ml to blood. If you don't get it, let me give you an idea. Okay, so we did this, right? We made this diagram, this one. So in this diagram, I said that systemic arteries carries oxygenated blood. Fine. What if I say that the blood that is coming here definitely is oxygenated blood, and it carries 20 ml oxygen per 100 ml of oxygenated blood. Is this wrong or right? If I say this blood is carrying 20 ml of oxygen per 100 ml of its sample, it's very true. Fine. And I say if this person is in the rest condition, the tissues of this person are not metabolically much active. These are tissues. So at this time, this person will deliver how many oxygen? 5 ml of oxygen to the tissues, and 15 ml will go straight as a part of deoxygenated blood. But same if I say the person is highly active, exercising, now tissues will get 15 ml of oxygen and 5 ml will go to the deoxygenated blood. This is what we say. Fine. Okay. So this is how the oxygen transport takes place. Let's talk about the transport of CO2 or carbon dioxide. So if I talk about carbon dioxide, 7%, like in oxygen, 3% of the oxygen was transporting in the form of 3% was traveling or transporting in the form of dissolved form in plasma. Here 7% is transporting in the dissolved form in plasma. Fine. Now 20 to 25%, how much? 20 to 25% goes with the hemoglobin. Again, here when oxygen was binding to hemoglobin, it was binding to heme portion. So it was known as oxyhemoglobin. But when CO2 binds to hemoglobin, it does not bind to the heme portion. It binds to the globin portion. So here hemoglobin plus CO2. Again reversible. Now this will not be known as carboxy. It will be known as carbaminohemoglobin. Why? Because this one is binding. This one is binding to which portion? To the globin portion. And globin is made up of amino acid. Now rest 70%, the maximum part, is transported as bicarbonate ions. Now what are these bicarbonate ions? Let's see. So whenever water and CO2 combines, they will form carbonic acid in the presence of the enzyme carbonic anhydrase. Which enzyme? Carbonic anhydrase. Fine. Whenever CO2 and water combines in the presence of enzyme carbonic anhydrase, which is present abundantly in RBC, so it will form acid, which is carbonic acid. Now with the help of same enzyme, I'm writing Ca for this enzyme. Okay. With the help of the same enzyme, this will dissociate into H positive ions plus bicarbonate ions. So this is what bicarbonate ions are, and 70% of CO2 is transported as such. Fine. All right. So let's solve these questions, guys. Nearly dash percent of CO2 is transported by RBC. RBC, whereas dash percent of it is carried as bicarbonate ions. So by RBC, they means hemoglobin. So 20 to 25% by hemoglobin and 70 as bicarbonate ions. So answer is 1. Next. Every 100 ml of deoxygenated blood delivers approximately dash ml of CO2 to the alveoli. So if we talk about how much ml of CO2 is transported by pulmonary artery to the alveoli per 100 ml of blood. I just listen, understand this thing. This is a line of NCRT. That's why I made a question out of it. So we say that every 100 ml of this blood gives 4 ml of CO2 to the alveoli. Okay, so how much ml? 4 ml. So it's a fact, you need to learn this, guys. Some things are factual and we need to learn. So that is answer 3. Okay, so 4 ml of CO2 goes from 100 ml of the deoxygenated blood to the alveoli. Okay, all right. Next. Factors that affect the diffusion of gases are thickness of diffusion membrane, very true. It affects solubility of gases, very true, and partial pressure. So answer will be 4, all of these. Okay, next. Transport of oxygen. Now let's talk about something which is very important, and that is your oxygen dissociation curve. So it's a topic under transport of gases only. So that's why I have written here heading, and that is oxygen dissociation curve. So whenever we have to understand the relationship, whenever, listen to me, because this is a topic where a lot of students get stressed out, but I tell you, this is a savior, and this is quite easy if you just get clicked in one go, right? So whenever I want to see the relationship between oxygen, it's oxygen's partial pressure, that means the level of oxygen, and the hemoglobin saturation, that means how much hemoglobin is carrying the oxygen. Or if I want to see the relationship between the two, for example, now you have a thing, you have hemoglobin and you have a machine by which you can increase or decrease the level of oxygen. So now you're going to see what will happen if I increase the level of oxygen. Hemoglobin will be saturated, but if I decrease the oxygen, hemoglobin will be empty. So this is what we see in this curve known as oxygen dissociation curve. Now, ma'am, you are saying that we are joining hemoglobin with oxygen, but why the name of a curve is dissociation? Because here we are more concerned about dissociation than the association. That's why its name is oxygen dissociation curve. How? You will get to know very shortly. So let's make the graph. Okay. All right. So sometimes, zoology people also make graphs. So this is your graph. Very lovely. Here we have partial pressure of oxygen. Here we have % saturation of hemoglobin. Okay. Now imagine you have hemoglobins. Okay. So when you increase the partial pressure of oxygen, the saturation starts like this and it will end up like this. So which type of a curve is formed here? S or S-shaped or sigmoid curve or sigmoid curve. Okay, so we say that here, what does sigmoid, wherever the words, the graph is sigmoid, it means that everything you start good, that means whenever, okay, so this is really going really bad, let me just make it again. Okay, guys, so whenever we say there is a sigmoid curve, that means whenever we are increasing the partial pressure of oxygen, whenever we are increasing the partial pressure of oxygen, the saturation increases. We increase more partial pressure of oxygen, more saturation increases. That means more hemoglobin is attached to the oxygen. But one time will come, the graph becomes stationary because all hemoglobin is saturated with oxygen. For example, if I say you have 15 gram of hemoglobin, you have how much? 15 gram of hemoglobin. If I increase the concentration of oxygen, imagine 5 gram of hemoglobin got saturated. Now, furthermore, more 5 gram got saturated. And then 15 gram got saturated. Now, the graph will be flat. Why? because the 15 gram of hemoglobin was the maximum amount and entire got saturated. Fine. Now, here the partial pressure of oxygen, as you can see here, the 100% saturation is there. Here 50% saturation is there. Fine. Wherever the 50% saturation is taking place, or at what value of partial pressure, the 50% saturation of hemoglobin is taking place, you call this value, for example, if this is 25 mm of mercury, I will call this value as P50 value. What is P50 value? I say P50 value is that partial pressure of oxygen where 50% of my hemoglobin is filled with the oxygen, 50% of my hemoglobin is saturated with the oxygen. Okay guys, right? All right. So here you can see the partial pressure is low and saturation is not completely done. What do you think? Where does this happens? This happens in your tissues. So this is the place where in tissues partial pressure of oxygen is less and this type of a graph is formed. And this where around 50% saturation has taken place is usually in your pulmonary artery. Okay, that means in deoxygenated blood, but 100% takes place in the oxygenated blood. So this will be the place of oxygenated blood. So if someone said, where does this type of a graph is forming? When your blood is deoxygenated, and this type of a graph will reach when your blood is highly oxygenated. So this is a criteria for oxygenated blood. This is a criteria for deoxygenated blood. Fine. Now this graph can shift towards left and right. What will be the difference? If I shift my graph to the right side a bit, then if I shift my graph to the right side a bit, then what will happen? My P50 value will increase. How? Let's see. See guys. Imagine I'm shifting my graph. Okay. Let me raise this for more space. You will get this slide. Okay. Don't worry. So here, if I say my graph moves to the right side, imagine such type of a graph is formed. So you can also see that the P50 value has been increased. This is the new P50 value now. Okay. Similarly, my graph may shift to the left side. Is this possible? Yes, practically possible. So this is the left shift. So two type of shifts occur. One is a left shift, another is a right shift. The right shift usually occur when there is increase in CO2. Always remember, CO2 is like your enemy and oxygen is like your friend. Okay, so left shift will occur whenever there is increase in oxygen. Okay, left shift occur whenever there is increase in partial pressure of oxygen, and right shift occur when there is increase in partial pressure of CO2. So this is bad, this is good. As you can see in this shift, the P50 value is less. So what does it signify? It signifies, imagine this is 10 mm of mercury and imagine this is 50 mm of mercury. Now you can see in just 10 mm of mercury, the entire hemoglobin is saturated. That's good for you. If in less money, imagine in 10 rupees, you are getting a big packet of chips. And second situation, in 50 rupees, you are getting the same packet of chips. What is better for you? In 10 rupees, you're getting the same packet of chips, right? So just like that, left shift is a good one. Left shift will allow the readily or fast association of oxygen with the hemoglobin. So this is always associated with, the left shift is always associated with the formation of oxyhemoglobin. And this is a bad one. It will make sure that your hemoglobin should not bind with the oxygen. So this is always related with dissociation of oxyhemoglobin, and this is always related with association of oxyhemoglobin, or the formation of oxyhemoglobin. Okay, now what are the factors that causes right shift? Let's talk about them first. Increase in partial pressure of CO2, increase in H positive ion, decrease in pH, increase in temperature, or increase in BPG. Whereas the left shift occurs whenever there is decrease in partial pressure of CO2, increase in partial pressure of oxygen, then there is decrease in H positive ions, just opposite to that. increase in pH, decrease in temperature, and decrease in BPG. So this will lead to the left shift. Left shift is usually, or this type of a graph is usually formed when you are talking about the oxygen and hemoglobin saturation near the alveoli, because there the partial pressure of oxygen is more and there is more association of hemoglobin. Okay, and this one occurs in the tissue. Right shift you can see near the tissues. Okay, for example, this is the normal sigmoid curve. You can see when you go near the tissues, this curve will start to shift towards the right. And near the alveoli, you will see the association of hemoglobin. In the alveoli, you see the association of hemoglobin and you will get the left shift there. And here in tissues, you will get to see the dissociation of the oxyhemoglobin. That's why if oxyhemoglobin will break, only then the oxygen will get to the tissues, right? For example, if I say in this diagram, the hemoglobin is coming in the form of oxyhemoglobin. If this get dissociated, only then the oxygen will enter here. Na? If hemoglobin will not leave the oxygen, how will it will enter inside the tissues? Now, how does hemoglobin leave the oxygen? We'll see it shortly. Okay. Now, so this is the oxygen dissociation curve. Now, why do CO2 H positive ions, they are causing the dissociation of oxyhemoglobin? Let's see. So whenever there is increase in CO2, CO2 will react with water and it will form H2CO3 and hence H positive ion and HCO3 negative ion. So that is directly proportional. Whenever you will get more partial pressure of CO2, H positive ion will directly increase. Why? Because it will react with this these molecules, CO2 and water will react and form H positive ion. So as a result, these H positive ions, these are enemies of oxyhemoglobin. So when these H positive ions, they see oxyhemoglobin anywhere, they will bind to it. And they will say, no, this is my friend, you go alone. This is my friend, you go alone. You go alone. This is what happens here. This is what happens here when the oxygenated blood is going or coming towards the tissues. When the oxygenated blood is coming towards the tissues, this is the criteria. That means what is happening here? HbO2 is coming here. But when at this point we saw that there is increase in CO2 concentration, when we see there is more CO2 concentration, CO2 will form H positive ion and H positive ion will lead to the breakage of the oxyhemoglobin. As a result, HBH will be formed here and oxygen will be released. This is what happens near the tissues. Okay, that's why it causes right shift. Now, what about temperature? Whenever there is increase in temperature, the globin structure gets destroyed. Globin, that means globin of hemoglobin, it will get destroyed. If it gets destroyed, will the entire structure will be intact? No. If the entire structure is not intact, how will oxygen bind with the hemoglobin? So that will lead to dissociation. So more the temperature, globin structure will be destroyed. If this structure is destroyed, guys, what will happen? Imagine this structure is completely destroyed. Hemoglobin will not bind to oxygen. Same with the BPG. BPG is biphosphoglycerate. Biphosphoglycerate or diphosphoglycerate. Okay. Biphosphoglycerate or diphosphoglycerate. This is a intermediate in the glycolysis. You know what is glycolysis? Breakdown of glucose. So see in the middle of that BPG or DPG, diphospho 2,3 BPG or 2,3 DPG. Di or bi same na. So that is formed. Okay, right? Sometimes I speak Hindi also. So biphosphoglycerate is formed, and when its concentration is more, it also destroys the structure of hemoglobin. This destroys the structure of hemoglobin. So this is how it will lead to dissociation of hemoglobin with the oxygen. All right, guys, that's about the shifts. Let's talk about the transport at two places. How does the transport take place? Imagine, now you will say, ma'am, you always say imagine. Now, after some time, you started saying imagine. Okay, nothing is possible without your imagination. So this is tissues, right? and this is your blood vessel. What do we have in blood vessel? RBC. RBC. Okay. Now imagine the blood that is coming here. The blood that is coming here is oxygenated. Which blood? Oxygenated blood. Just the way we have done here, guys. Just the way we have done here. I'm just talking about this portion again. I'm talking about this portion again. Okay. Don't worry. So here in tissues, because oxidation is taking place, so there will be more production of CO2. It is producing CO2. Now, this CO2, 7% will be dissolved in plasma, right? Around 23% will bind with the hemoglobin and form carbaminohemoglobin, and 70%, how much percent? 70% will react with water and form carbonic acid, and hence HCO3 negative plus H positive ion. This is what we have done here also. This is what we have done here also. Just look at it. Hurry up. Now, because this blood is oxygenated blood, so the oxygen is already saturated with the hemoglobin. But when this blood reaches the tissue is producing a lot of CO2. So CO2 will produce H positive ion. And this H positive ion cannot bear these two lovebirds together, Hb and O2. So this H positive ion, what will they do? They will bind with this one, causes the dissociation of oxygen, and oxygen will lead to or go to the tissues. So what's basically happening here? We are saying where at this position there is increase in partial pressure of CO2. The more CO2 you will produce, it will lead to the more dissociation of oxyhemoglobin. Hence, it is leading to the transport of oxygen or unloading of oxygen. Imagine hemoglobin was a truck and this truck was loaded with oxygen. Now it has came to the site where we need construction. Okay, for example, this oxygen is your sand or bricks, and hemoglobin is truck, and this is a site of construction. So now this truck is taking the oxygen, right, or this truck is taking the bricks. Now when H positive ion came, what is H positive ion? Imagine these are something. For example, you are unloading the bricks here and you're picking up the cement from here. So now H positive ion is cement and oxygen was a brick. So you are unloading the bricks and you're loading the cement now. This is what we are doing. Okay, so by increase in partial pressure of CO2, it leads to unloading and transport of oxygen. This effect is known as Bohr's effect. What do you call it as? Bohr's effect. In Bohr's you have O na, and this leads to the transport of O. Bohr's effect. Now this same is going to the alveoli. Whatever is formed is going to the alveoli. In alveoli, the partial pressure of oxygen is more. Okay, and this is the endothelium. And this is the RBC. This RBC is coming and it is a part of deoxygenated blood. Okay, now what will happen? What have we produced here? One thing we have produced here is HbCO2. HbCO2. This is one thing we have produced. Another thing we have produced in this one is HbH. These two things we have produced, and we are bringing this in the RBC as these forms. Now when H positive ion is formed here, what about HCO3 negative? HCO3 negative will move back in the plasma and react with sodium or potassium ion, and it will act as buffer. What does it act as? Buffer. Okay, now in place of HCO3 negative, chloride ions enter here, and you call this phenomenon as chloride shift. What do you call it as? Chloride shift. Okay, now what will happen when this RBC, which has a lot of carbaminohemoglobin and hemoglobin bind with the H positive ion, when it will enter around the alveoli. Okay, it's moving, the blood is moving. Now we are talking about this portion, guys. Now we are talking about this portion, this one, this portion. Okay, here we have made HbH and we have made what? HbCO2. Now this blood is moving and it has reached here. So here the blood is having HbH and HbCO2. How does CO2 will move there? Let's see. So what happened, because there is increase in partial pressure of oxygen, due to increase in partial pressure of oxygen here, this entire will become HbO2, and CO2 will be unloaded. CO2 will be unloaded because now the partial pressure of oxygen is more. Also, because the partial pressure of oxygen is more, again the reverse takes place. HbO2 will form and H positive ions will be free. Now because H positive ions are free, this HCO3 negative will move inside. HCO3 negative will move inside and chloride ions will move back. And this is known as reverse of chloride shift. This is known as reverse of chloride shift. Okay, because H positive ion is now free. Now HCO3 negative is a die-hard lover of H positive ion. HCO3 negative, though H positive ion already betrayed HCO3 negative and bind with the hemoglobin. But now HCO3 negative is a die-hard lover. So when he saw that H positive ion is free, so HCO3 negative will come back again and reverse will take place with the help of same enzyme, which is carbonic anhydrase. And what will be formed, guys, here? What will be formed? H2O plus CO2, and CO2 will go in the alveoli, and CO2 will go in the alveoli. The enzyme will be same. That's why we say it's a reversible process. So this is how, when near the alveoli there is increase in partial pressure of O2, which leads to unloading or transport of CO2, you call this effect, which is quite opposite to the Bohr's effect, is Haldane's effect. What effect is this, guys? Haldane's effect. Okay. So this is how transport takes place. Now you must be thinking, ma'am, so tough. Nothing is tough. If you know how the gases are transported, it is easier. And if you know near the tissues the partial pressure of CO2 is more, it is easier. Just if you, I know this is a kind of a thing you will not understand in one go. What do you have to do? Rewind it. Watch it again. Okay. Don't worry. Stay calm. Don't worry. Okay. All right. So this is how the transport of gases takes place, guys. Let's talk about regulation of respiration. So there must be something that leads to the, regulation that can control the respiration. So for controlling the respiration, we have two things. One are the centers and other are chemosensitive areas. Chemo means chemical. These areas are sensitive to chemicals. That means they can detect your chemicals. They can detect your chemicals. First, we'll talk about centers. Centers are main. So centers, they are of two types. One is respiratory rhythm center. So centers, also known as respiratory centers, they are of two types. One is respiratory rhythm center and another is pneumotaxic center. Both these centers, as everything is controlled by brain, so both these centers, they are present in the brain. Respiratory rhythm center is present in the medulla oblongata and pneumotaxic is present in pons, because in pneumotaxic there is P. So P for pons, right? Because in exam you will get a written thing. No, nobody is going to speak. So there you can see P, pons. Otherwise when you speak the P is silent. So this one controls normal inspiration. Normal breathing, just like now you are doing the breathing now. It is only leading it whenever the medulla is giving signal, you inspire for certain seconds. So when the signal is off from the medulla, the muscles will not contract and naturally expiration takes place. So signal is always sent for inspiration, because in normal inspiration muscles are contracted. We always have to send the neural signal when we need to contract the muscle. Right? Imagine if I say in normal inspiration, two muscles are contracting. In normal expiration, no muscles contract. So when medulla is giving signal to your external intercostal and diaphragm muscle, they contract and leads to inhalation. Imagine that occurs for 0.4 seconds. Now after 0.4 seconds, the medulla will stop giving signal and naturally the muscles will relax and expiration takes place. This is what happens here. The pneumotaxic center is known as switch off center. So you must be thinking it will stop your breathing. No, switch off doesn't mean stop your breathing. It alters the rate of breathing. Alters the rate of breathing. How? Imagine while normally you are inhaling, imagine you inhale for 0.4 second and you exhale for 0.4 second. So while you are running, your breathing rate gets increased, it faster, so it gets fast. So what happened during at that time, you're not taking deep breath, you're taking shallow breath. For example, now you will breathe for 0.4 in spite of 0.4. You will inhale for 0.2 second. That's why our breathing is like that. Okay, but while we are normally inhaling deep breath, it's like that. Here we are inhaling for more duration. So altering the rate means more shallow breaths. More shallow, but more in number breaths. Okay, so this is how it alters the rate. In chemosensitive areas, we have two types of chemosensitive areas. One is central, another is peripheral. So both these areas, they can detect certain chemicals. These are they have chemoreceptors. What do they have? They have chemoreceptors. So these chemoreceptors are present in the brain as well as in the blood vessel. So the one which are present in the brain, you call them as central chemoreceptors. You call them as central chemoreceptors. They are present in medulla near this center, respiratory rhythm center. Whereas peripheral, they are present in blood vessel like carotid artery and aorta. You all know what is aorta? The one that takes away the oxygenated blood from the left ventricle. And carotid artery is the artery present over here and it supplies blood to the brain. Fine. So all these chemosensitive area, they can only detect partial pressure of CO2 and H positive ion. Increase in concentration, mind it, okay? So for example, in blood vessel, imagine in blood vessel, there are certain chemoreceptors present here. Okay, now imagine the partial pressure of CO2 increases. These chemoreceptors can readily detect the increase in partial pressure of CO2 or H positive ion. They cannot detect the increase in partial pressure of oxygen. First of all, they will always detect the increase, not the decrease. They always detect the things in higher concentration. They cannot detect oxygen. The reason is because oxygen is present in conjugated form with the hemoglobin. 97% is already bound to the hemoglobin. Free is only 3%, and chemosensitive areas can only detect 3% of the oxygen, which is very low. Or chemosensitive areas can only detect free gas. It cannot detect the gas which is in the bound form. So here no oxygen can be detected. It can only detect the partial pressure of CO2 and H positive ions. Fine guys, that's about the regulation. Let's talk about the certain terminologies. So whenever we use the word hypoxia, what does it mean? It means decrease in partial pressure of oxygen. If I talk about hypercapnia, what does it mean? It means increase in partial pressure of CO2. Okay, so when does hypercapnia takes place? When there is no exchange of gases. Or when does hypoxia takes place? When there is no exchange of gases. Hypoxia can take place by various factors. One such factor can be altitude sickness. What is altitude sickness? I'll let you know shortly. Second, it can be due to anemia. Imagine you do not have enough RBCs. No RBC, no hemoglobin, no oxygen, right? It can also occur due to toxicity. Imagine some toxin came into your body and it binds to hemoglobin or it destroys hemoglobin. It can lead to, right? So these are certain factors which can lead to hypoxia in your body. All right. Okay. Let's move further and talk about the disorders. Now, why did I explain you hypoxia? Because that will be important in disorders. All right, my dear students. First is asthma. You must have heard of asthma. In English, we say asthma. In Hindi, we say asthma. Okay. So, asthma is difficulty in breathing and there is wheezing sound due to inflammation of bronchi and bronchioles. What happened in asthma? It's basically the allergy or hypersensitivity. A person is allergic to dust. Person is allergic to dust. As a result, the person's bronchioles or bronchi, they got inflamed. So when these bronchi and bronchioles get inflamed, there is narrowing of lumen. Lumen means the inner space. For example, you can say this is a normal lumen so wide, right? The inner space, it can easily lead to the transport of number of gases or large amount of gas. But in asthma, the person's lumen get narrower like this, so less oxygen enters, and person bronchi or trachea will start producing sounds, wheezing sound, like that. Okay, it's not even like that, it's a wheezing, very sharp sound is like that. Okay, so that leads to difficulty in breathing, and a person can even die due to asthmatic attack. That's why that people always carry that pump. What is that for? That is for dilating the lumen. That is for dilating the lumen. Okay, all right. Next is emphysema. Emphysema is a chronic disorder. Chronic means very deadly, in which alveolar walls are damaged, due to which respiratory surface is decreased. One of the major cause of this is cigarette smoking. So you can see this is a lung of a smoker and this is a lung of a normal person. So when a person smokes so much, the alveoli gets damaged. Alveoli gets damaged. So when alveoli gets damaged, the surface area for exchange of gases, it decreases, and no more gases can enter inside the body, and this can be fatal. A person can even die due to this disorder, that is emphysema. So remember, this do not affect upper respiratory tract, it affects lower respiratory tract, that is your alveoli. Okay, next let's talk about occupational respiratory disorder. What are these? So occupational respiratory disorder, occupation means work, place. So person works at a place where the person is exposed to certain chemicals every day. For example, a person is working in the glass factory. So person is exposed to silica every day, and that leads to silicosis. Or a person is working in a factory where asbestos is formed, and person gets the disease. The person gets a disease, asbestosis. What is asbestos? In many homes, you must have seen that green color shed or blue color shed. It's generally they are made for shade or preventing the sun rays entering directly into the house. So that is made up of asbestos, right? So for example, a person is working there and cutting that sheet. Those fibers from the sheet will enter inside the nose and it gets into the lungs. In the lungs, they are present the trachea and bronchi. Okay, now what will happen? The wall of the bronchi, it contains elastic fiber. Which fibers? Elastic fiber. Imagine these are elastic fibers. Okay, so when these chemicals, now let's make chemicals. When these chemicals, okay, what color should I use? Red. When these chemicals enter inside this, elastic fibers are destroyed. And when these fibers are destroyed, you call it as fibrosis, which leads to the increase in the thickness. When the muscles are destroyed, they are no more elastic, the thickness of the wall increases. The thickness of the wall increases, hence narrowing of lumen. The fibrosis, it leads to thickening of wall. Thickening of wall. Why? Because earlier the this fiber was elastic and it was single. Now it has been broken down into pieces and no more elastic, which will lead to the accumulation of small parts, and hence the inflammation response start and the wall become thick, leading to narrowing of the lumen. Okay, so by fibrosis there is thickening of wall occurs. You call it as proliferation. Okay, let me just read this for you. So when the thickness increases, there is narrowing of lumen and a person is not able to breathe. This can lead to permanent damage also. So that's why people who are working in these industries, they should be provided with proper mask and all. Okay, this can also seen in people and the diseases, black lung disease, in the people who work in coal mines. In coal mines. Okay, all right. So in certain industries, especially those involving grinding or stone breaking, so much dust is produced that the defense mechanism of the body cannot fully cope with the situation. Now earlier, when you have a 1 or 2 exposure with the dust, usually you can also go to factory and you get exposed, but you will not get a disease. The constant exposure will cause a disease. Why? Because when they come in a small amount, our immune system can easily cope with them and they can destroy them. Okay, but when there is a constant exposure, long exposure give rise to inflammation leading to fibrosis, proliferation of the fibrous tissue. Proliferation means growth. It will get thickened and thus causes serious lung damage. Workers in such industry should wear protective masks. So people should wear masks that will protect them from that long exposure. Okay. All right. So this was about your occupational lung disorder. Next is pneumonia. So pneumonia is a disease that occurs either through a bacteria, which is Haemophilus, or Streptococcus. Okay, what are these bacteria? So the bacteria enter through droplet infection, what you call it as sneezing or coughing. So when bacteria enters inside your alveoli, obviously a bacteria is entering. The mucus cell will secrete a lot of mucus. As you can see, there is a lot of fluid here, fluid in the alveoli. We say that in pneumonia, the fluid accumulation occurs in case of pneumonia in the alveoli. So from where does this fluid come? First, this fluid is basically the mucus. The excess mucus production occurs whenever there is inflammation. For example, during cold, you get infected with virus, right? And you get a lot of runny nose. Just imagine the same thing occurs inside the alveoli. So it will lead to accumulation of fluid. Also, this fluid comes from extracellular fluid. That means the fluid present outside the tissues. Because the virus or the bacteria, right? In case here, the bacteria will kill these cells, leading to the entry of fluid here. So this basically contains extracellular fluid and mucus. So whenever there is fluid is filled in the alveoli, this will decrease the surface area for exchange of gases. There will be decreased surface area for exchange of gases. Okay, so this is what happened in pneumonia. So people who have extreme pneumonia, their in fact fingernails and skin can become blue. Why? Because because there is no exchange of gases, so a person will face hypoxia and hypercapnia. And due to hypercapnia, what is hypercapnia? Increase in CO2. Due to hypercapnia, the blood will appear blue, right? You call it as cyanosis. Cyanosis is blue blood. We always draw the deoxygenated blood as blue, and veins are blue because they carry deoxygenated blood, because more CO2 will give a blue color. And whenever we are talking about blue in biology, we use the word cyano. Okay? All right. Next, let's talk about carbon monoxide poisoning. So what happened in carbon monoxide poisoning? Though hemoglobin bind to the oxygen and form oxyhemoglobin, but when there is, burning of a fossil fuel, imagine there is a room and you burn a fossil fuel there and there is no window, no ventilation. Carbon monoxide will be formed. Carbon monoxide is formed by burning of fossil fuels. So that carbon monoxide is very much poisonous because it has affinity towards oxygen around 200 times more. It has the affinity towards the hemoglobin than the oxygen around 200 times more. See, if I say oxygen is binding with hemoglobin, their love is okay, eternal and so. But when CO comes, carbon monoxide, the hemoglobin and CO, they have love from generations. Or what do you say in Hindi? Janmo janmo ka pyar, right? So CO has 200 times more affinity than oxygen. So this will bind to, when this binds, the process is irreversible, and now no oxygen can bind, hemoglobin is completely saturated, and a person will eventually die of suffocation. So this happen in carbon monoxide poisoning. So wherever there is more pollution, there is more carbon monoxide, and person usually have these breathing issues, or sometimes they feel dizziness, more dizziness, right? Wherever there is increase in pollution. When I was living in Delhi, I know there was a lot of pollution. At a point of time, there was a lot of pollution that I always used to feel dizzy and my eyes are irritating. Why? Because my hemoglobin is saturated with carbon monoxide. So how my oxygen is going to be there in the tissues? It will be difficult for that. Okay. All right. Okay. So let's talk about mountain sickness. What about mountain sickness? So whenever you go at high altitude, like in mountains of like Mount Everest or Rohtang Pass or Ladakh region, at high altitude there is less partial pressure of oxygen. Okay, imagine earlier at the plane, I'm just giving you a rough value, the partial pressure of oxygen was 5 mm of mercury in the atmosphere, and in the lung it was 0. So how much is the difference? 5. A lot. The gases can easily pass. Imagine at high altitude, this drops to 1 mm of mercury, and this is again 0. So now tell me, how the gases can move? No, there is a less partial pressure in the gases. So whenever there is less partial pressure of oxygen in the atmosphere, no or less exchange takes place. Exchange of gases takes place. So that's why whosoever goes to high altitude, especially in Ladakh and so, they are advised to rest for 1 or 2 days and get acclimatized. So what happens when you do not have enough oxygen? You will feel nausea, a feel of vomiting, right? You will, get an extreme headache. And sometimes it can even lead to fatal condition. A person needs oxygen cylinder, you can see that person is using. Okay, so that's why always stay for 1 or 2 days, have rest, and then started going for tourism and all, right? Like a tourist. So whenever you go to high altitude, these are the things you face. But those people who already are living, they get acclimatized. Acclimatize means adapted. So how does body gets adapted, or even after 1 or 2 days? Your body, it will increase the number of RBC. It will increase the number of RBC. More the RBC, more oxygen transport. Less the RBC, less oxygen transport. If oxygen is less increase the truck. That's it. Okay. So this is your mountain sickness. Let's talk about decompression sickness. So mountain sickness was when you go at high altitude. So when you go at low altitude, or you go in the diving. So when you go for diving, there is increase in partial pressure of gases. Increase in partial pressure of gases. And which gas I'm talking about here is nitrogen. So when a person, see that person has undergone for diving and now he's feeling decompression sickness. So what happened? For example, this is a surface of water and this is a seabed. This is a seabed. Okay, now diver goes down. So when the diver goes down, there is increase in partial pressure of nitrogen. Earlier, the partial pressure of nitrogen was less. So that's why the partial pressure of nitrogen was less. So that gas was not able to enter inside my body. But now, the partial pressure of nitrogen has increased down here. Now, nitrogen enters. And this will lead to dizziness. Right? So that's why it is advised not to stay for around more than 45 minutes down in this seabed. Even I have done the diving. I have seen the corals in Maldives, right? So it's really beautiful. But even I have suffered some decompression sickness. I'll let you know shortly. So now when you have to move up, it is always advised to move in this manner, like this, in this manner. A person should never move up in one go. Why? Because when you move up in one go, the nitrogen leaves tissues and it will enter blood. And when it will enter blood, it forms bubbles. It forms bubbles. So when a bubble is formed, the capillaries got burst. Capillaries burst, and this can lead to a lot of disorders. Sometimes the capillaries of this eyes get burst and the eye got serious injuries. Fine. So when you go up, you should go in a zigzag manner. So these symptoms of dizziness is usually seen by, or it can, it's a common symptom for every diver. So when I went for diving and I went up for 2 days, my body was like that. I was not able to walk. For example, you have a fever but you do not have high temperature, and you're not able to walk, and you're dizzy, like your entire body is paining, it's aching very much, and you do not have, like in Hindi we say na, jaan hi nahi rehti, just like that. Okay. So this is what happened in the diving, but it's really beautiful. Whenever you get the time, and you do not fear of, you have no fear of water, and you have that courage, definitely do it in life. It's a, once in a life thing. It's worth the shot. So the chronic respiratory disorder due to heavy smoking in which the alveolar walls are damaged is asthma, bronchitis, pneumonia, emphysema. Simple. Cigarette smoking, emphysema. So now you must be thinking, ma'am, have gone to Maldives and for diving. And so, when I was in my bachelor's, and our teacher was teaching us about these ciliary traits in detail. So ciliary traits, they secrete calcium and they form corals. And she's shown us one that video, and I was so much fascinated. So I told my professor that one day I'll collect the money and I will definitely go. And that was somewhere in my mind. One day I will do something, I'll do a job and I will collect all the money, because my parents was not able to afford such kind of a big trip, right? So, and I will definitely go for diving. And yes, I did, and I'm really proud of myself. I was so much fearful. I was not able to jump from that boat, but I made it. I tell you, I made it. And once I was inside that water and touching those corals, amazing. That was amazing. I think you should also make some goals in life and fulfill them. This is what it's all about. Life is all about this. More goals you have, more happily you will live, and you will never get bored of yourself, and you will always be doing something, always be doing something. All right, guys, next. Lack of pulmonary surfactant can cause asthma, atelectasis, cystic fibrosis, emphysema. So if imagine I don't have surfactant. What is surfactant? Lecithin. Surfactant is something that decreases surface tension. That decreases surface tension. If the surface tension, or I say the surface tension increases, or lecithin is less, then what will happen? Collapse. And other name for collapse is atelectasis. This is the another name for collapse, right? Next. Which of the following is occupational lung disorder? Anthracis? No. Anthrax is anthracis only, and this occurs to us. Anthrax to animals, anthracis occurs to us due to Bacillus anthracis. Okay. Silicosis is the occupational respiratory disorder. What is botulism? Botulism is a kind of disorder that occurs due to bacterium Clostridium botulinum. It leads to paralysis of muscle, botulinum. This is a bacteria. Okay. It causes paralysis of skeletal muscles. Next. Okay. Pneumotaxic center can moderate the function of the respiratory rhythm center. It is present in. So simple question, where is pneumotaxic center present? You say 2. The another line was there just to confuse you. So this is what I have also seen in the exam of NEET 2022. They have written a lot of lines just to confuse you. But you are the tigers and the tigress. You do not get confused. Next. High PO2, maybe that is not visible, but this is high partial pressure of oxygen. Low PCO2, lesser H positive concentration, lower temperature. It is saying high PO2, low PCO2, low H positive ion, and low temperature. So all these causes left shift. Now read the option. Favorable for oxygen hemoglobin dissociation and causes right shift? No. Forget all those which have right in them, right shift in them. Okay, next. Favorable for formation of oxyhemoglobin and causes left shift. Left shift is good and oxygen will bind to hemoglobin. Answer is 3. In 4th, it is written dissociation of oxyhemoglobin. So this is incorrect. Next. So next, is thank you. So this was all about this chapter. I hope you will study it well and read the NCRT also. And I'll bring another lecture, another chapter, and I want you guys to please read NCRT side by side, because that's really, really, really very important. And mark all the PYQs and whatever, booklets or modules you have for practice. And I wish you all the very best. I'll meet in the next class. Bye bye. Take care. Lots of love.